Okay, again, welcome everyone. It's the top of the hour. We have a good number of people here. This is Marie with ILSI North America, and we just wanted to welcome you. We might have some more beeps as we go along, John, but we'll just kind of ignore those. Those are people just popping on a little bit late. So as usual, we won't go through introductions of everyone on the call. I think we did that the first time and it took 15 minutes. So I would just like to provide an introduction here. So again, welcome. This is a webinar organized by the Gut Microbiome Committee of ILSI North America. And today we have Dr. Jonathan Swann with us, who is an associate professor in microbiomics and human development in the Department of Surgery and Cancer at Imperial College London. Dr. Swann is responsible for leading metabonomic-based research programs to understand the influence of gene-environment interactions on the mammalian metabolic system and their implications for health and disease. One of his primary research themes just happens to be the role of the gut microbiota in the mammalian metabolic system and their associations with health and disease. And this includes their contribution to host endogenous and xenobiotic metabolism, the gut-brain axis and its role in neurological disorders, the carcinogenic potential of the microbiome, and environmental and host factors that shape the acquisition and the composition of the gut microbiome. Dr. Swann's research is supported by the Bill and Melinda Gates Foundation, AstraZeneca, the National Sorry, John, I think we got a little feedback there, among other groups. He's also on the editorial board for the journal Microbiome, and he sits on numerous expert panels relating to the gut microbiota. And one of those was part of ILSI Europe. So today we'll hear more about some of the work that was undertaken by ILSI Europe, and this project was introduced to the committee actually in 2018, early on in 2018. So this is the outcome of that work. So with that, I would like to turn it over to Dr. Swann.
Great, thanks, Marie. Yeah, so as Marie just said, I'm going to go through some of the outcomes of this expert group put together by ILSI Europe. So hopefully, I'm not entirely sure the makeup of the audience to this call, but hopefully most people are aware that the body is colonized by a huge array of microorganisms, and it's becoming more and more apparent that they have a strong influence to affect human health and disease. And a kind of key aspect of this is that they are accessible to dietary interventions. We can manipulate them, and the idea we can manipulate these microbes through food, through nutritional interventions to actually result in health outcomes. And so we're interested in whether we can change the microbiome to go from, say, this population to this population, probably unlikely, but also the problem when it comes to microbiome research and trying to understand it all is that it's a rapidly developing and emerging field. And so the populations in which a lot of interventions are studied vary wildly. And just because an intervention works in one population doesn't necessarily mean it's going to work in another. On top of that, there are also a huge array of tools that we can use to study the gut microbiota. We can measure who's there, we can measure what they're doing, we can measure how the host's responding to these. And these new tools are getting more and more sophisticated and complex, producing greater and greater amounts of data. And the way we can interpret that data and handle that data is also, there's lots of different ways. So different groups will do the data analysis in different ways. So it's often hard to actually take some of the findings from certain publications and try and translate them to a wider population. And so that was sort of some of the motivation behind this task force. Can we provide guidance to also try to interpret this data? In addition, one size doesn't fit all. Like a probiotic that works in, say, an infant isn't necessarily going to work in an adult, or one that works in an obese population might not work in a lean population. And if we don't do this carefully, we can result in outcomes like this was a headline in the BBC last year. Probiotics were labeled quite useless. And there was actually a really good response to this by Gregor Reed. And here Gregor just explained how using the wrong probiotic or a probiotic in the wrong population can result in these kind of outcomes. And we have to be really careful in how we communicate these findings. And so this kind of all provided this motivation to this expert group that was put together by OC Europe. And so, yeah, like I say, this was, I guess, twofold. One, to provide guidance on designing and conducting human studies for looking at how foods can influence the microbiota and human health, but also, yeah, how to interpret the results that are being published. And so we broke this down into these main areas: the study design, eligibility criteria, how we characterize the food products, how we measure what these impacts are doing, and also some legislative aspects as well. So in terms of study design, there's already a wealth of really good papers out there outlining guidance for design, conduct, and reporting of human intervention studies. So it wasn't really our aim. Our aim was more to highlight key areas that are relevant to the gut microbiome. And so when you're designing your study, it's really important that one of the first things you get out there is where you define your research question. This is going to form your the basis for your study hypothesis as well. So what is it you're actually trying to test or study? Where's the knowledge gap? And we highlighted some potential areas of relevance to the microbiome, and these aren't exhaustive, and there's also a lot of overlap, but just some areas to think about is what effect does the gut microbiome have on the metabolism and the bioavailability of nutrients and non-nutrients in foods? What effects do these foods have on the gut microbial composition or activity? And what effect do changes in the microbiome following these dietary interventions have on human health? And so this is a kind of good starting point. And when it comes to study designs for studying the microbiome, one problem we have is that a lot of the microbiota research so far has all been based on, or has largely been based on case control studies. So comparing, for example, lean versus obese individuals. And this has been useful, but it only really provides correlations and associations. It doesn't tell you information about the cause and effect. And so instead, randomized control trials would be a more preferred, and intervention trials provide a much better way of getting at these kind of cause-effect relationships. And so randomized controls trials is the most rigorous type to achieve this, particularly studying foods. And there's a number of different randomized controlled trials that can be performed, and the most sort of gold standard would be the double-blind placebo-controlled RCTs. And so the two main ones used in microbiome research would be the parallel or the crossover designs of these RCTs. So just for anyone unfamiliar, the parallel trial would be where you had a study population, and you split them into different arms. This would be the parallel, and they all receive the different treatments, either placebo or treatment, all in parallel to one another. The alternative is the crossover design, where each individual will take the placebo, but there'll be a washout period, and then they'll be put on the treatment, or they'll be randomized to have the treatment first, washout period, then placebo. So every single individual has both placebo and the product. As well as parallel and crossover trials, though, initial exploratory studies can also be performed. These can be single-arm studies, and these can be useful just for gaining useful information. So things such as identifying an effective dose for dose finding, also to measure the variability and size of the response to the intervention. These can be useful for these, and this can be necessary information in order to perform sample size calculations. Also, the persistence, or how long does the effect of the intervention last for, the persistence of the effects, and this can be really useful for understanding carryover effects, and this is necessary in order to design crossover studies. So just to highlight some main advantages and disadvantages to these two different types, so whether to use crossover or parallel. Crossover designs have the benefit of each individual being part of the treatment and the placebo group, and so the variance at baseline is tighter in the crossover design, whereas in the parallel arm, the variance might in the microbiome might be unbalanced between the two different treatment arms. And as a result, the sample size can be smaller for the crossover design and but larger for the parallel. But the parallel arm design benefits from having shorter durations, and this means that participants don't have to give as much time up, and so there's more likely that there's going to be less requirements, there's less dropout, and whereas crossovers have longer durations and potentially more likelihood of dropout. Use of facilities and resources, though, the crossover design, this is more stretched out over time, so it's less demanding on a particular, less focused on a particular time point, and so it's more evenly distributed, whereas not for the parallel. Also, the data analysis for the crossover design, because you have repeated measures, the same person measured at multiple time points, the data analysis is more complex as well than the parallel arm. And also there's this idea of crossover effect. If you don't know how long the effect lasts for, then you can't do the crossover design. It has to be a parallel design. And crossover designs can also be useful for identifying responders and non-responders. So this was a paper published in 2014. And this was looking at 3 different diets: a weight loss diet, a non-starch polysaccharide diet, and a resistant starch diet. And here, each number is an individual put on the different diets, and this is changes in the microbiota composition. And what you see is those in this circle here, they have a low response to the diets. Their microbiota doesn't change very much in response to the diets, whereas those outside of the circle, they have a larger response to the diets. And if this was a parallel study, you wouldn't perhaps see that they all responded sort of didn't respond to the diets in the same way. So this non-responder status would not be identified from a parallel approach. You can also see, looking at the run-in diets, that the non-responders actually had a higher diversity, whereas the responders had a lower diversity. And this is something we see quite often, that initial diversity can explain this response, more resilient to the changes. So if you've got your study design, then you want to decide who is going to be enrolled. And so eligibility criteria, these are physiological and clinical characteristics or demographic variables that can be used to define the study population. And in particular, what we're interested in are factors that might modify the microbiota. And it's important that we control, or at least are aware of these, because these could lead to false positive or false negative results. Now, some of the most important eligibility criteria are the age range of the participants. So we know that there are at least 3 distinct age ranges. There's the infant, where the microbiome is rapidly accumulating, it's very diverse, it's diversifying. There's the adult, the infant is less diverse, but is developing, and the adult is more stable and diversity, characterized by diversity, and then the elderly microbiota, which is losing diversity. And in particular for the infants, we know that health is characterized by low diversity, so as in it's usually dominated, or in a health perspective, it's dominated by bifidobacteria. So a dietary intervention targeted at infants will be different to that targeted at adults. And then there's a lot of debate about when the infant microbiota stabilizes onto an adult-like microbiome. So, for example, in this paper, they identified that by 3 years of age, the infant microbiota is adult-like, whereas in this paper, they identified that adolescent children have a different microbiota from adults. So again, this is something to be aware of where your age cutoffs lie. And when it comes to the infants as well, things that are important to consider are things like delivery mode, gestational age, breastfed versus formula fed, weaning age. These are all factors that we know can influence the microbiota. And if it's not possible to restrict your population based on these factors, it's important to capture this information for your data analysis. diet is obviously a huge impact on the microbiota, and this appropriate information should be collected and control should be applied to try and control your study population on this sense. In particular, supplements included in the diet that might influence the microbiota are important to be restricted. Particularly fiber is a good example of this. And this was a study published 2018, last year, showing that habitual fiber intake could influence the results or the response to the prebiotic, an inulin type prebiotic. Here, what they demonstrated was individuals who have a high fiber habitual diet, 39 grams a day, compared to those with a low fiber habitual diet, change their responsiveness to the inulin. You can see here that, so the darker bars here are those with a low fiber diet. And what you'll see is those on a low fiber diet are much more responsive to this prebiotic. And so there's some suggestions that you should try and capture habitual fiber intake information on your participants before starting the study, perhaps even recruit based on this. It's also been shown that weight loss and calorie restriction can modify the microbiota. So individuals known to be undergoing weight loss diets should be excluded from the study, unless, of course, this is part of the research question. And the same applies to specific diets like the vegan diet or like those on gluten-free diets. Again, this all comes down to a research question, but if this isn't part of your research question, then including vegans into your study may sort of confound the results. And so in terms of assessing dietary intake, this is often done through diet diaries. And this should be done throughout the study. So at the beginning of the study, the end of the study, and then throughout to check that there are no drastic differences in the diet which might cause a change in the microbiota that's unrelated to the intervention. Now, diet diaries are notoriously subjective and subject to bias, but they can be useful. And then there's also objective biomarkers starting to emerge to measure metabolites, say in urine, that can predict dietary intake. There's also information emerging that there are sex differences between the microbiota. Now, again, these next kind of examples, it's unlikely that you would control your study population for these because then it starts becoming so unique to a particular group of people that maybe the intervention is no longer widely applicable. But these are important considerations, like in terms of data analysis. You might want to model the females separately from the males. Also evidence that exercise can impact the microbiota. So individuals that are going to change their exercise routines may be excluded from the study, or that should be captured. And then also things like smoking and lifestyle factors that are known to impact the microbiome should be captured as well in your metadata. And then we have the environmental considerations, things like your living environment, so crowding, family composition, family size. These are all important, particularly in early life. It's been shown that family members share bacterial strains, and that shows transmission amongst family members. It's been shown that the number of family members you have positively correlates with the number of bifidobacteria species present in the gut. Household pets are also a rich source for the microbiota and strong influences. It's been shown that infants with dogs have more ruminococcus and oscillospora than those without. Also, hygiene practices and cleanliness of houses. Now, this is quite hard to capture, but again, as much information as you can capture on the study population can help when it comes to the data analysis. And so it's obvious then that there is going to, even if you control for a lot of these factors, there's still going to be a significant amount of variation in the microbiota across your study participants. And there's even ideas now about sort of rethinking the approach and actually using this to your advantage, this known variation at the start. And there's even ideas now about using the microbiota status in potential inclusion exclusion criteria. Now, obviously this all depends on your ability to sample the participants, collect the data, analyze the data, and then go back and decide which participants are going to be involved in the study. It's probably not that valid at this point in time, but going forwards, this may be an attractive proposition. Or perhaps measuring just a specific bacterial group. If you know a specific bacterial group is involved in your mechanism of action, then this could be in advance. Marie, can you still hear me? I've got an error message.
Yeah, I can hear you loud and clear.
Sorry, I just got a connection issue. Okay. So then in terms of defining sample size, this is a question we get asked a lot, and really the microbiota is not a great target to address sample size with. And this is because when it comes to measuring the microbiome in a sort of untargeted manner, so you're just blindly measuring the microbes present in a fecal sample, say, you can't pre-select which bacteria you expect to change with the intervention. So in some ways, you haven't got a hypothesis to test by measuring the bacteria. It's also not possible to define what is a normal microbiota or even what's an abnormal microbiota. So you haven't really got a handle to base this on. Also, it's very difficult to interpret whether an increase or decrease in a specific bacterial group is a sort of marker of any kind of health effect. And so really, unless you've got a very targeted bacterial questions, for example, you're testing the bifidogenic effect of a fiber, in which case you can just measure the bifidobacteria with, say, qPCR and then use that to calculate your sample size. It's more effective really to base sample sizes on a different outcome measure. So say, for example, if you're interested in how a probiotic could lower blood cholesterol via the microbiota, then the blood cholesterol levels will be more useful to measure sample sizes with. In terms of characterization of food products, a product should be well characterized in order to help with health claims. In general, this should include the macronutrient composition, so the fat, carbohydrate, and protein content, also fiber content as well, and any other aspects that might modulate the microbiota. These should be defined within your product. When it comes to probiotics, before in vivo determination of the probiotic properties, the candidate strain should be fully characterized. This includes things like carbohydrate fermentation, enzymatic activity, the ability to survive and grow under GI conditions. And this should be done at the species and strain level as well, because a lot of these effects can be strain specific. Strains should also be named in accordance with the International Code of Nomenclature, and they should be deposited in an internationally recognized culture collection. Their safety should be demonstrated, and also information on their viability and stability, particularly for things like storage as well, and the expected dose, the minimum dose, should be well characterized. And then there are some examples where the viability might not be an issue. So, for example, Akkermansia muciniphila. It's recently been shown that the pasteurized form of this, so when it's been killed, can actually have greater effects than when you give an equal amount of the viable cells. And this is due to the membrane proteins of this particular bacteria being recognized by toll-like receptors and inducing its response that way. In terms of prebiotics, you've got to demonstrate that a prebiotic is resistant to digestion. Now, a lot of the assays that perform this are not always accurate, and so digestibility assessments might also need to be made in mice and humans. But for prebiotics, the source of the fiber, the carbohydrate composition, degree of polymerization, average molecular weight, and glycosidic linkages should be clearly defined as well. And polyphenols are emerging now as more and more microbial modulators. This is because they're poorly absorbed in the upper gut, passing to the large intestine, where they can modulate the microbiome and result in health benefits. And in terms of polyphenols, then the source of polyphenol should be clearly determined. The chemical structure should all be characterized. The amount of the polyphenol in the intervention should also be ascertained, and often this is quite highly underestimated. And this is because a lot of the polyphenols present in the matrix are often not measured. And a lot of these can be what we call non-extractable polyphenols, so they're not released by solvents when they're typically tested. And then the placebo that's used, so the match to the test product in terms of sensory characteristics, the appearance, its energy content and administration, it needs to have a minimal effect on the microbiota or its activity. And when it comes to prebiotics, some common ones are maltodextrin, which is frequently used in prebiotic interventions, but there is some evidence that when used at high doses, maltodextrin can also alter microbiota composition. An alternative is microcrystalline cellulose, which can be used, it has minimal energy, and it is less fermented by the gut microbiota. And then the validity and relevance of any intervention study depends on a high level of participant compliance. So by that we mean the degree to which the subject adheres to the study protocol. So any factors that affect compliance should be considered, and to try and maximize the protocol related compliance. So far, there's been little information on how sampling aversion affects compliance. There was one study, the Flemish Gut Flora project, that found of all their participants, only around 5% considered quitting due to sampling their feces. And of those, even 2.8% of those thought about quitting because of storing the feces in their freezers. So this doesn't seem to have a huge effect on compliance. And one of the major factors in this study they found was that providing very clear instructions on how to collect the samples and store the samples was part of the reason why the individuals were happy to maintain on the study. Also, methods to measure and optimize compliance should be implemented. So, for example, biomarkers can be good to get an objective measure of compliance. So things like probiotics, you can actually detect the probiotic strains in the feces. This provides a good measure of compliance. Fermentable fibers, things such as short-chain fatty acids and fecal pH can also be used. And then the metabolites of polyphenols can be measured in blood, urine, and feces to kind of gain an understanding of how much the participants are complying with the protocol. So acceptable levels of compliance should be decided before the study starts, and any major deviation from this should result in them being excluded from the trial. So in order to determine how whether the intervention has had an effect, there's a whole variety of different endpoints that can be measured. So just to introduce some terminology here, largely what we're measuring in a lot of these microbiome studies are the bacteria present. In theory, we should be measuring the fungi, the viruses, the protozoa, but largely when things like 16S sequencing is performed, it's the bacteria that are being targeted. And when we measure who's there, this is referred to as metataxonomics. So this is 16S sequencing to determine who's there. All these individual bacteria bring with them different genomes, which bring with them different genes. And when you think about all these genes collectively, it's what referred to as the metagenome. And so when we measure all of these genes together, this is what referred to as metagenomics. These genes can then be transcribed into messenger RNA transcripts. So these genes can be expressed into messenger RNAs. And when you consider all of these bacterial messenger RNAs, this is the metatranscriptome, which can be measured through metatranscriptomics. These can then be translated into proteins, forming the metaproteome, which can be measured by metaproteomics. Generally speaking, metatranscriptomics and metaproteomics are not routinely done, and certainly not routinely done for intervention studies. But as we move forward, they might be more and more applied. But these are very intensive computationally, as is metagenomics. But these proteins then give rise to these proteins, these enzymes perform metabolic functions which give rise to metabolites. And technically speaking, this should be the meta-metabolome, but refer to this as the metabolome, and this can be measured through metabolomics or metabonomics, or metabolic profiling. And these metabolites then can have a direct effect in the gut, so they can bind to receptors that line the gut, or can influence gut health or metabolism in the gut and digestion. They can also be absorbed from the gut into the blood, where they can be circulated around the system, and they can have more systemic effect, including effects on, say, the brain. In addition, the bacteria can have a direct effect on the gut and on the body through things like the immune system, through activation of the immune system as well. And this can lead to health outcomes, so phenotypic outcomes, and these can also be measured. So there's lots of ways we can measure how the intervention can act on the bacteria in the gut to trigger all this cascade and result in health outcomes. In terms of measuring the effects on the bacteria themselves, the main methods traditionally or typically used is 16S rRNA gene sequencing or metataxonomics. So here, differences in a particular gene is measured in the feces, and bacteria that are taxonomically similar have more similar composition of this gene, whereas those are more different have a different composition of this gene. And this can be used to bucket the sequences into what we call OTUs, or operational taxonomic units, which can then be assigned confidently down to the genus level. So we can measure which genera are present in the feces and how abundant they are. Can't confidently go down to species and strain level. Like I said, this tells you who's there, and then metagenomics measures the complete genes present in the feces, and this tells us what the functions or potential functions of that community can do. We can also take that information and go down to the species and strain level. So metataxonomics 16S tends to be a lot easier to do than metagenomics. It's also about a factor of 10 cheaper than metagenomics. Other more traditional approaches can be things like fish analysis, qPCR, microarrays. qPCR is very good for very targeted analysis of bacterial groups. So again, if you're interested in, say, the lactobacilli, you can measure them through this qPCR approach. So when it comes to sampling considerations, things that are important to remember, it's emerging things like stool consistency can have effect on microbiota richness. So this might need to be taken into account when sampling. Time of day can also have an influence on the microbiota composition. So if possible, sampling should be tried to normalize the time of sampling across the individuals. Also, things like freeze-thaw cycles. Freeze-thaw cycles can increase the risk of bacterial cells lysing, and so when they lyse, they release their DNA. So then if you pellet your cells before doing the DNA extraction, you would lose that cell-free DNA, and that bacterial group would be underestimated. So again, it's something to be taken into account. So you can use this bacterial information in a number of ways. People can calculate alpha diversity, which tells you things like richness, number of different bacterial species there, evenness, the relative abundance of the different taxa. Beta diversity tells you things like heterogeneity of the microbiota, so how comparable they are from one to another. Say, for example, with this particular index, if you have a score of 0, tells you that the two communities are identical, whereas if you get a 1, it shows those two communities are not composite. There's no compositional overlap at all. You can also measure relative individual taxa. And so diversity is good because diversity in an ecosystem is good generally, as it improves the stability of that ecosystem. It helps to build in things like metabolic redundancy. So that means if you lose a particular individual from the community, from the ecosystem, the overall network wouldn't fall apart. Also, by having this high amount of diversity, also increases the ability to respond to environmental challenges and stresses. So high diversity is generally considered a good thing. This is not the case in infants, but for adults, high diversity is generally good. And it's been shown through a number of studies that loss of diversity has been associated with various diseases like immune diseases, metabolic diseases, cancer, and autism. But it's been shown that this was a meta-analysis on 64 human studies exploring dietary fibers, and there was no positive association between dietary fiber and alpha diversity. But there was in the fiber was able to increase the abundance of the bifidobacteria, lactobacilli, and fecal butyrate. And fecal butyrate, we'll talk about in a minute, has been associated with a lot of health benefits. So alpha diversity isn't the only thing to measure. And in fact, if your intervention was to increase or boost a particular bacterial group, and say the bifidobacteria, and increase their dominance in the profile, diversity would actually go down if you increase them. So alpha diversity isn't always the only thing or best thing to measure. And actually, measuring specific bacterial groups may be more informative. So in this study, it was shown that a beneficial response to kernel-based bread, which lowered blood glucose, this beneficial effect was associated with the Prevotella. Also targeting specific bacterial groups. Like I say, there's conflicting data about the benefits of lactobacilli and bifidobacteria. Generally, these are considered healthy to be boosted, but for example, lactobacilli has been shown to be increased in individuals with obesity, but has also been shown to increase weight loss in obese individuals. So there's conflicting data. Butyrate producing bacteria have also been shown to be beneficial. And in particular, Faecalibacterium has been shown to, in Crohn's disease, has been shown to have anti-inflammatory properties. So if you've got a very targeted and known mechanism of action, targeting a particular bacterial group can be useful. Now there's what we would call metabolic redundancy or functional redundancy in the microbiome. So different bacterial species may do the same function. They may produce the same metabolites. So compositionally, even though they look different, if they're performing the same function, does it matter? And so one way we can get at this kind of question is by studying the metabolites. And we do this through metabonomics. So metabonomics, we tend to measure urine, blood, or fecal samples or tissue biopsies. And here, these are technically they're host samples. So if you're looking in urine, for example, you're getting metabolic information on endogenous metabolism, so the metabolism that's encoded in the genome, but you can also pick up metabolites that come from environmental inputs. So you can pick up metabolites from your dietary component, perhaps even from your intervention, and you can also pick up metabolites that come from the intestinal microbiota. So you can get a functional readout of what these microbes are doing. You can also identify metabolites and measure metabolites that come from interactions between these things. So you get a metabolic overview of how all these different components are interacting metabolically. And we often get asked, what's the best sample type to measure? Fecal samples can be good because they tell you information directly about what the microbes are doing in the gut and how they're interacting with the diet. But technically speaking, a lot of the metabolites we see in the feces, they haven't been absorbed from the host gut. So they haven't really seen the host metabolic system. So how relevant are fecal metabolites is up for debate. Whereas urine metabolites, for example, have been absorbed from the gut, have interacted with the metabolic system, and have then been excreted. So you get some information on this host microbiota interaction as well. Blood metabolites tell you information about the metabolites that are circulating throughout the body, but generally speaking, there has to be quite an insult to the body in order to drastically change the circulating plasma metabolites. And it's been found that a lot of microbial metabolites that arise from ingested foods, they appear in the blood after only 5 to 7 hours, and most are cleared by the liver and the kidneys after about 12 to 8 hours. So this is important to bear in mind when collecting the blood samples for studying these microbial metabolites. And around the fecal microbiota was able to explain about 68% of the variance in the fecal metabolome, showing that intimate association between the microbial composition and the fecal metabolome. So in terms of the metabolites we can detect, these endpoints from the microbiota Short chain fatty acids are probably one of the best studied. These have been associated with a huge range of health benefits, things like gut health, implicated in appetite regulation, anti-cancer properties, a whole range of different properties. The bile acids are also important. Bile acids are synthesized from cholesterol in the liver to make the primary bile acids. These are conjugated with glycine and taurine. For being concentrated in the gallbladder. When you consume a meal, they're released into the small intestine where they help digest fats and vitamins and help in their absorption. And around 95% of these bile acids are reabsorbed, recycled to the liver, but around 5% spill into the colon where they're broken down by the microbes. First of all, they're deconjugated. They take off the glycine and taurine, and then they convert them into what we call secondary bile acids, which can then be reabsorbed, returned to the liver, and then recycled again. And it's been shown that these bile acids can bind to receptors in the gut and also throughout the body, throughout the liver, the brain, the kidneys, the heart, and they can bind to these receptors and actually act as signaling molecules to regulate metabolism. And different bile acids or different microbial produced bile acids have different signaling effects. So this is one way through which the microbes can influence host metabolism. And then a whole range of other microbial metabolites can also bind to these receptors, both throughout the gut and throughout the body. Tryptophan metabolism by the microbiota is another important aspect of this. So tryptophan is a dietary amino acid, comes from a lot of meat intake, and the microbes can break this down into indoles, and indoles can bind to receptors that are a hydrocarbon receptor and can have beneficial effects on immune status, like immune homeostasis. But also these indole can be sulfated in the liver and can actually act as a uremic toxin, act as a toxin in the kidneys. Can also have both neurotoxic and neuroprotective effects on the brain, depending on its metabolism. So this kind of demonstrates nicely the both positive and negative aspects of these microbial metabolism. And so there's a whole range of different metabolites produced by the microbes or substrate for the microbiota that have been implicated in a whole variety of both beneficial and negative health outcomes. Generally speaking, carbohydrate metabolism fermentation by the microbes results in things like short-chain fatty acids, succinate, which tend to have more health benefits, whereas metabolites that come from amino acid fermentation, like paracresol and paracresol sulfate, these tend to be associated with more negative outcomes. And then there are some, it's like a kind of Goldilocks syndrome, where just enough is right. They have immunoregulatory effects, but if you have too much, then can result in things like inflammation and genotoxicity. And then we have other health outcomes like immunological aspects. So we know that the microbiota can influence the immune system, contributes to immune effector cell activity and maturation. Dysbiosis in the microbiota has been associated with autoimmune diseases like IBD. And we know that the microbes can influence the immune system both through direct contact with immune cells, particularly at the gut level, and they can also produce signaling molecules like short-chain fatty acids and bile acids, which can also have an effect on the immune system. And so ways in which we can measure this immune interaction or immune activation by the microbes, we can measure things like cytokines, chemokines, we can measure gene expression of things like pattern recognition molecules, receptors. We can also, so these tend to be measured by ELISAs and multiplex immunoassays. qPCR can be used to measure things like toll-like receptors and nod-like receptors. In terms of the sample types to study this, then, peripheral blood samples provide information on the systemic immune effects, whereas fecal samples may be more informative about the immune effects occurring in the colonic environment. But it's important to remember that the fecal samples won't tell you much about what's occurring in the mucosal tissue. And so to get that, you probably need a biopsy sample. And for an intervention study, this is unlikely to get this. And there's been some good studies then trying to put all these together. So this was one demonstrating that here they measured serum immune markers, fecal immune markers, alterations in the fecal bacteria, and then also changes in the short-chain fatty acids and other microbial metabolites, kind of demonstrating how you can use this multi-omic approach to study this. And then it's important to remember this was a meta-analysis on 42 randomized controlled trials of over 2,000 adults, showing that probiotics could significantly reduce serum levels of pro-inflammatory cytokines and increase the anti-inflammatory cytokine IL-10. Whereas when this meta-analysis on 10 randomized controlled trials from elderly individuals, it was shown that there was no effect of the probiotic on inflammatory responses. And this is possibly due to the different immune environments of these two study populations, with elderly individuals likely to be undergoing immunosenescence, and so their immune status and this dialogue with the microbiota is likely to be different. And so when it comes to health outcomes and what do you measure, this is largely research question specific and depends on what you're trying to actually explore. So this could include things like appetite status, this could include BMI, blood pressure, traveler's diarrhea, cognitive measures. Like I say, it all depends on your research question. And just some examples here. This was a meta-analysis showing that probiotics could significantly reduce body weight, BMI, and fat percentage. And also could significantly reduce blood pressure. So then just finally, legislative aspects. This has mainly been written from an EU perspective rather than a US perspective, and so likely to be different across the countries, across the continents. And so for EFSA, from a European point of view, more than 3,000 health claims were evaluated, but the majority were not considered scientifically substantiated. And the reasons for a large amount of these being rejected is due to insufficient characterization of the ingredients, lack of demonstration of a claimed effect that was beneficial to health, lack of demonstration of a cause and effect relationship. And I think this is one of the hardest things to pull apart in microbiota intervention studies, and also lack of support from high quality pertinent studies. So with that, that's kind of the main aspects of the work we've been doing. It's important to just emphasize finally that this is a rapidly evolving field, and as more and more studies, more and more information is gathered, that eligibility criteria is likely to change. We'd like to understand more and more about what is affecting the microbiome, what can influence it, and also this methodology and the endpoints we can measure are likely to develop further and further and refine the different endpoints that we measure. So I'll leave it there, and I'm happy to take